Thank you so much, Shane. Hello, everyone. Welcome to the November instance of our Catalyze webinar series. I'm very excited to introduce today's topic, which will cover the scientific priority areas for research and funding among the divisions that make up the National Heart, Lung, and Blood Institute, which Catalyze is supported by. So we have 4 presenters here today representing each of these divisions of NHLBI, who will share the scientific priorities of their division. And at this time, I'd like to pass the virtual speaking baton to Dr. Mike Pieck, the Scientific Director of the Catalyze program, to give a brief introduction before introducing each of today's presenters.
Thanks, Marissa, and thanks again to our panelists here from my colleagues from across the NHLBI. So the goal today is for you, the Catalyze community, to hear directly from the different divisions and kind of understand a little bit more about what their specific missions are, and in particular, what are their specific priority areas where new innovations might be helpful for applying for the Catalyze program. So just very briefly, we have Denny Buxton from the Division of Cardiovascular Sciences, Patricia Noel from the Division of Lung Diseases, Asif Rizwan from the Division of Blood Research and Resources, Blood Disease and Resources, and George Mensah, who is the director and one of our senior executives for the Center for Translation, Implementation, and Science. So appreciate you all taking the time today to spend time with us. Next slide. So just to orient and remind everyone, so the Catalyze program kind of supports this stage in between basic research discovery and in preparation for clinical trial research. And in particular today, we're going to be going over the product definition, RFAs, the funding announcements. Next slide. I think we highlight them in the next slide. And in addition, as a reminder, we're still under development for the preclinical program, and hopefully we'll be able to have some good news in early 2023. I'll hand this along. So next slide, please. So just as a reminder, there are 5 RFAs that make up the product definition bucket. And these include the enabling technology, the small molecules and biologics, and device and diagnostics. And as a reminder, the next application deadline is November 21st. And if you were hoping for a more detailed description of the program and the ins and outs of applying, we did that webinar in July. So if we go to the next slide, I think there's a hyperlink and a reference to that webinar where you can learn more about the details of the special requirements and also some of the services available through the Catalyze Coordinating Center. But I did want to highlight one reminder that we'll be posting to the website, and that's the next slide. So in feedback for some of the applicants, we've developed these checklists that are specific for each of the different RFA types. So these will be available as a downloadable PDF for each if you're applying for one of the specific RFAs, but they'll also be part of this the slide deck after the presentation. The one thing I do want to note is the update to the Enabling Technology Transformer Platform, HL23-010. There is no longer a requirement for a separate attachment for your milestones. Those should now be part of your research proposal. And this is really important because if you add it as an attachment, it'll be kicked out for administrative reasons. And with this, I would like to introduce our first speaker. I think. Next slide, please. So first, we're going to hear from Denny Buxton from the Division of Cardiovascular Sciences. Denny.
Thank you, Mike. And good morning, everyone, or good afternoon, depending on where you are. If I could have the next slide, please. So the Division of Cardiovascular Sciences, or DCVS, is the largest of the extramural research divisions, and it covers a very wide range of science, from basic science and early translation through clinical trials to population sciences. The division is broken down into 7 branches: advanced technologies and surgery, vascular biology and hypertension, atherothrombosis and coronary artery disease, heart development and structural diseases, heart failure and arrhythmias, epidemiology, and clinical applications. And I've grouped the 7 branches into their programs. So advanced technologies and surgery and vascular biology and hypertension are part of the basic and early translational research program. The next 3 branches are in the adult and pediatric cardiac research program. And then finally, epidemiology and clinical applications in the Population and Prevention Sciences program. So these 7 branches encompass something in the order of 100 program officers, clinical trial specialists, and medical officers. And so what I'm going to present in terms of priorities is very much a sort of abbreviated and potted list, because each one of those 100 staff will have their own priorities. Some of the priority areas are derived from recent working groups, workshops that we've held, and the recommendations that those groups have put together, and other come from discussions within the branches. So if I could have the next slide, please. So some of the priority areas are related to our surgical group. So the development of heart valves suitable for use in low resource environments is one priority. Rheumatic heart disease is still a major problem in the less rich countries, and so diagnostics and therapeutics for rheumatic heart disease are an area that we are very interested in. Therapeutic extracellular vesicles have demonstrated potential in preclinical and a few clinical studies, but we feel that there's a need to improve and optimize them for efficacy and reproducibility. Biomaterials and biomimetic coatings tend to be designed on a fairly uniform system rather than being designed specifically for the site where they're going to be implanted. And so our technology staff feel that there is an important area for tailored biomaterials that would match more closely the cellular milieu at the planned implantation site. 2 or 3 of our branches came up with improved methods for measurement of ambulatory blood pressure. This is really something that I think would be of tremendous benefit in our clinical trials, and also in the real world for maintaining proper blood pressure in people without white coat effects. We've recently had a couple of working groups on lymphatic disorders, and there's a definite consensus that we need better diagnostics and therapeutic modalities for lymphatics. This is an area of science that has really been underappreciated and under-researched, and we would really like to see more in this area. Next slide, please. Peripheral arterial disease is a serious problem for a growing number of patients, particularly as the number of diabetics increases. And so we'd like to have improved methods for monitoring blood flow in these patients. Similarly, enhanced visualization of small blood vessels for preclinical and clinical brain imaging would be a useful step forward. Hypertension remains a major problem, and we'd like to see advances here in terms of cross-correlation of biomarker data and electronic health records, and early identification and better monitoring using machine learning and AI technology. Measuring arterial stiffness is a fairly cumbersome process at the moment, and so we would like to see wearable devices that can measure multiple cardiovascular outputs and to help with this process. Preeclampsia remains a major problem. Maternal morbidity and mortality in this country is quite shameful, really, and preeclampsia is a significant contributor to that mortality and morbidity and mortality, and we really don't have good therapeutics for preeclampsia or good diagnostics for early diagnosis, and so that's another area. And improved diagnostics and treatments for aortic aneurysms. At the moment, diagnosis of aortic aneurysms is based largely on watching and looking at the size, and so we would like to have an improved diagnosis of who is going to progress in their aortic aneurysms and how to treat them. Next slide, please. Turning now to the atherosclerosis side of things, we would like to see effective vaccines that target PCSK9 and reduce LDL-C, and like to have proof of principle studies using these vaccines in animal models. We'd also like to see vaccines targeting other molecules in lipid metabolism like ApoC3 and ANGPTL3 and 4, and how these can help to control triglyceride levels. Artificial intelligence, I think, has a role to improve diagnosis of ischemia-producing obstructive coronary stenosis on CT angiography. And finally, lipoprotein A is a risk factor for coronary artery disease that is poorly understood. And so we'd like to see reliable assays that are isoform independent using a monoclonal antibody. So those are all the areas that I wanted to talk about. But as I mentioned before, this is very much just a sort of pot shot of a few of the areas. And we have interest in a much wider range of science. So if you have an idea that isn't on this list, we'd like you to contact DCVS staff to discuss it with them and discuss where, what our priorities for that would be. So thank you for your attention.
Thank you, Denny. And just as a reminder, you can type Q&A questions into the Q&A box as the speakers are speaking, and then at the end we'll have a broader discussion across the division representatives. All right, so next we're going to hear from the Division of Lung Diseases from Patricia Noel. Patricia.
Thank you, Mike, and hello to everyone, and thank you for joining. If there's a problem with the volume at any point, too low, too loud, please let me know and I'll try to adjust. I wanted to take the first couple of minutes to try and set the stage a bit for the Division of Lung Diseases approach to translational research. It really began, I would say, intensely in the early 2000s for several different reasons, not only the well-described chasm between the discovery of novel mechanistic insights when it comes to pulmonary biology and pathobiology and the translation into clinical application. I mean, that's a well-known problem that plagues all fields, but also the understanding that has been enhanced over the years that expression of disease may not be reflective of the pathobiology. So in other words, there may be many patients that have similar symptoms in a given disease, or even across disease areas sometimes, but they may have very different pathobiology. And really trying to understand that and treat the disease process rather than end-stage symptoms began to drive the division's interest in trying to provide translational programs for our research, different research fields. May I have the next slide? And so this, I want to start with the organization of the division. Dr. Jim Kiley is our director. We are divided into two pulmonary branches, the lung biology and disease branch. You can see that we cover acute and critical care, lung transplant, rare diseases, pulmonary response to infection, HIV, microbiome. And then we also have neonatal and pediatric diseases, as well as developmental and regenerative biology. In the airways biology and disease branch, we have asthma, CF, COPD and environment, women's health, genetics and advanced technologies, digital health and informatics and integrative lung biology, And then under the lung division organizational structure, we have the National Center on Sleep Disorders Research, which is led by Dr. Mariska Brown. And their concentration is on sleep disorders medicine, prevention and sleep health, neurobiology and sleep, and chronobiology. And really, you know, this is the formal structure of our division, but I'm sure you can tell just by looking at the areas, there's a lot of crosstalk. There is, these slides are going to be made available and posted. There is a link in the lower right-hand corner that if you click on it, will take you to the NHLBI page. That will describe these areas in more detail and also give you program staff contacts. Next slide, please. So this slide goes to one of our early programs in which we were using our standard funding mechanisms, in this case, the program project grant, to try and provide and encourage our investigators to take basic science findings and really try and take a pragmatic approach with them and really push towards clinical application. And this, and it was really meant to be bi-directional, where the basic science would contribute to the development of clinical application, and then clinical studies would go back and inform the basic research. And this particular example comes from a program project called the Asthma Inflammation Research Program, headed by Dr. Serpil Erzurum at the Cleveland Clinic. And the overarching goals were the role of chronic inflammation and repeated and failed repair and resolution cycles in asthma, and the development of novel diagnostics, treatments, and monitoring tools based on pathobiology. Again, that concept of trying to develop diagnostics, treatments, and management strategies based on disease biology. And this was, you know, one of our very early efforts that didn't really have a ton of structure or support built into it. You know, we tried to be as helpful and as flexible as we could. And this was actually a very successful program. We are currently testing one of the products developed in this program project in one of our clinical trials networks. But this program, again, is an example, but as we moved along, and as the Institute moved along and began developing multiple programs in the translational space, we really wanted to try and think about, you know, what are some guiding principles that can help us really effectively facilitate this space for our communities. Next slide, please. And again, for DLD, and I think, you know, many of my colleagues would agree with most of these, we came up with sort of several foundational principles. So we really wanted programs that have flexible support across the research continuum. I think everybody here is familiar with the idea that, you know, it may sound really good on paper, but getting there are going to be a lot of twists and turns. We need multiple types of support in training efforts beyond our traditional mechanisms, because we're not just talking about hypothesis-driven research that much of our investigative community is familiar with. We're talking about IP, we're talking about commercialization and regulatory, and certainly we have communities and people in the audience that are familiar with these concepts. But in the beginning, when we were really trying to bring our investigative research communities along, you know, these are areas where there was not a lot of experience, a lot of familiarity for many people. And then for the lung division in particular, we were interested and are interested in disease modifying interventions for targeted patient populations. And that will often require the integration of diverse expertise and data, as well as standardization of data and biospecimens. And then, of course, these types of programs require continued evaluation and refinement because we want to continue to enhance our efficiency and success of these complex translational programs. Next slide, please. And so I think that, we have come to Catalyze both as an institute in the hopes that we are integrating these concepts and can really provide a flexible funding and support continuum that goes from product definition all the way through preclinical development, and that we can provide this across the spectrum of technologies, drugs, biologics, small molecules, as well as devices. Next slide, please. And I want to give you a few examples before I go into our priority areas of some active projects that DLD has right now that are supported and funded through Catalyze. One of them is the mitigation of bronchoconstrictive effects of airway smooth muscle in asthma. And this project is really targeted at the development of a small molecule inhibitor to abrogate the tethering of the airway smooth muscle to surrounding matrix. And if this can be done successfully, that will help to reduce bronchoconstriction. Another one is a respiratory stimulant for use in critical care settings. So another small molecule, a novel class of small molecules, but this is not necessarily disease specific the way the asthma technology is. And so I'm just trying to give you some ideas across the spectrum. This is for respiratory stimulation without blunting the effects of analgesia. So really important in the ICU for patients that they are having a difficult time coming off the vent. And actually it's successful. The hope is that may also be helpful in diseases where patients may have a limited respiratory reserve, but still need analgesia. And then lastly, a device, bedside diagnostic imaging, mobile digital tomosynthesis for high resolution imaging at the ICU bedside. So the picture shown there is not from that project, it's just generic, as are the others. On the right-hand side, it's a DGT image as opposed to a standard X-ray on the left-hand side. And so this is meant for more clarity, obviously, in terms of diagnosis, but also there's often a lot of machinery, so to speak, and tools and tubing and things like that in the way. And the mobile DGT can obviously be taken to the bedside, but also deal with all the imaging, the tools that might be in the way of obtaining clear images and provide higher resolution even without those. So these are some examples. The next slide shows some of our priority areas. Again, as my colleague, Dr. Buxton said, these are just examples. So please reach out and be in touch. Again, our priority is for disease-modifying technologies and products for prevention and treatment of acute and chronic lung diseases. For example, novel mucolytic drugs. We know that the mucus involved in the pathobiology of different chronic lung diseases is different in composition than normal healthy mucus in the lung. And so novel mucolytic drugs that can actually help to thin and clear pathobiologic mucus are very important. Formulations to overcome mucus barriers. This speaks to delivery, as well as formulations to deliver uniform dosing to distal airways. Modulation of innate and adaptive pulmonary defenses against infection. And here I'd like to draw a distinction between a technology that may be aimed at the infective agent. That in general would not fall under the Division of Lung Diseases purview. If you need to be directing a technology to the pulmonary defense against the infective agent, and that would fall under our purview. Again, happy to discuss. Novel therapeutics, along with companion diagnostics, if appropriate, for disease-specific inflammatory repair and remodeling processes, and novel therapeutics targeting extrapulmonary processes that may contribute to development or worsening of lung and sleep disorders. So, for example, metabolic abnormalities. We have a number of lung diseases, and there are sleep disorders in which we know abnormal metabolism is contributing to worsening of the disease. And then we have digital health and informatics technologies, climate change related exposure and impact on lung and sleep disorders. Again, not just to measure the level of particles, pollutants, what have you in the air, but to actually look at it in the context of the lung and sleep disorders. Detection, predicting, and monitoring lung and sleep-related disorders during pregnancy and the postpartum period, an understudied problem. Again, as Dr. Buxton referred to, safe, accurate, and effective devices for use in neonatal, perinatal, and pediatric care settings. Validity and reliability of general purpose computing technologies for diagnosis or management of lung and sleep conditions. Evaluation of image pattern recognition tools for the detection and diagnosis of lung disease, again, speaks to the need for high resolution and mobile technologies. And lastly, mobile health technologies and electronic health records to target healthcare provider and or patient behaviors and interactions for promoting adherence to evidence-based guidelines for lung and or sleep health. So I know that was fast and a lot, but again, examples only. We encourage you to reach out to staff in the lung division. We are always happy to talk to people about the best way to facilitate the translation of your discoveries. And thank you very much, Mike.
Thank you, Patricia. And as a reminder, we have the NHLBI Catalyze at nih.gov email. So if you have any questions about where your technology fits, or if you need to identify who the program officer is, the Catalyze program can help with that. So next, we're going to hear from Asif Rizwan from the Division of Blood Diseases and Resources. Asif.
Thank you, Mike. Can you? All right. Yeah, I can see the slide. Yes, I'm Asif Rizwan. I'm a program officer and a program director in the blood division. The Division of Blood Disease and Resources, DBDR, is a leader in research on the cause, prevention, and treatment of non-neoplastic blood disease. Blood division also has a major responsibility to ensure the safety of the nation's blood supply. We are divided into 3 branches: blood epidemiology and clinical therapeutics branch, molecular, cellular, and system blood science branch, and translational blood science and resource branch. Together, we manage blood research activities at NHLBI and promote communication across the division, NHLBI, NIH, and other partner agencies. Epidemiology and clinical therapeutic branch supports epidemiologic, clinical, and implementation research throughout the spectrum of blood science. Molecular, cellular, and system blood science manage discovery science with a focus on the physiology and pathophysiology of the blood, bone marrow, and blood vessels. Translational branch supports therapeutic development and manufacturing, discovery science from bench to first-in-human studies, and opportunities in small business programs. Dr. Julie Panepinto is our acting division director. You can follow her on the Twitter that is given on the left side here, and you can read the blood-related news in our website as well. Next slide, please, Mike. All right. Blood disease affects millions of people each year in the United States. Blood disease and disorder impact the normal biology of red cell and white blood cell, platelets, bone marrow, vascular endothelium, and plasma proteins. Blood disease and disorder can present a wide range of symptoms. All of these have created a need for preventive, therapeutic, and curative approach for restoring health. In DBDR, we support research on benign blood disease, gene therapies, cell therapies such as hematopoietic stem cell transplants, transfusion medicine, research to understand the critical role of blood, bone marrow, vascular endothelium. We also support stimulation of post-discovery science, preclinical research, and commercialization initiatives. Next slide, please. So here are some examples of research that we cover. I tried to divide them into 3 categories based on Catalyze funding opportunity announcement, device, small molecule and biologics, and enabling technologies. These are these are only examples. For more information, please feel free to reach out to us. Device, point of care diagnostics device to personalize treatment. Point of care test for beta thalassemia carrier, diagnosis of sepsis. Microfluidic diagnostics such as microfluidic for neutropenia detection, microfluidics to study sickle red blood cell. Paper-based device, finger stick device for hematology and cytometry analysis. Wearable sensor for disease such as chronic monitoring of, say, venous thromboembolism. AI, machine learning powered devices such as microclot imaging device, blood disorder diagnosis using machine learning driven technology, intravascular sonothrombolysis, continuous monitoring or measurement of clotting and coagulation, imaging technology to visualize the hematopoietic stem cell in their niche. So these are some examples of device. On the small molecule and biologics, we have interest in therapeutics for sickle cell disease, small molecules to treat degenerative blood disease, extracellular vesicles for sepsis, development of platelet-like particles. In the small molecule, we also have interest in vascular and hematopoietic regeneration, treatment of iron overload, safe anticoagulant and antithrombotic. For example, inhibition of factor 11 system, nuclear targeting small molecules and biologic, to increase the stem cell expansion. That's an example. Hemostasis treatment, small molecule that could help people with hemophilia A and B, development of tolerogenic factor VIII to prevent inhibitor development in hemophilia A, small molecule integrin activators to enhance blood transplant, antibody therapy for human hematopoietic regeneration, anti-thromboinflammatory agents, targeted delivery of clotting factors. So these are some examples of small molecules and biologics. Enabling technology, we support research on single-cell proteomics of blood cells, stem cell expansion, biomanufacturing of hematopoietic stem cells, biomarker discovery, technology for improving transfusion efficiency, like research in transfusion medicine, blood storage, gene editing therapy for hemoglobinopathies, sickle cell disease, neovascularization of engineered tissue, platelet-like particles, like engineered particles, for the management of coagulation disorder, gut microbiome technology to study human gut microbiome, technology to study glycan structure, ultrasound-mediated thrombolysis, maternal health. So these are some examples of enabling technology. As the previous speakers have mentioned, like, these are only some examples. If you have an abstract, if you have an idea, please feel free to reach out to Catalyze leadership. Feel free to reach out to us, like, any one of us who works in the NHLBI in different division. I will be happy to help you. And also, I would highly recommend to visit NIH Reporter website. From Reporter website, you will get an idea what types of grants are funded within NHLBI. And if you have any other additional questions, please feel free to reach out to me. Thank you. Thank you so much.
Thank you, Asif. So we've heard about the heart division, the lung division, and the blood division. And it's my pleasure now to introduce Dr. George Mensah, the director of the Center for Translational Research and Implemented Science, which, kind of like Catalyze, covers the whole institute. So George, please take it away.
Thanks very much, Mike. If you could give me feedback in case the volume is okay. Excellent. Thank you. Greetings, everyone. And I should begin by letting you know how proud we are of the Catalyze program. And I think everything you've heard from my colleagues is true. We would do everything possible to provide you the technical assistance for you to write outstanding applications. In fact, I should also add that the bulk of the scientific and technical information you need to write the best applications you've already heard. So I have a very, very simple message to conclude this round. Is for your outstanding, creative, innovative ideas to really have transformative change and transformative impact, it will also be very good to think of some of the implementation barriers. Even though the real bulk of the work you want to do is to be able to overcome the valley of death, to overcome many of the challenges in early translation. But the message here is, as you put together your team, also begin to think of some of the priority areas that I'm going to share with you. But the truth is, everything you've heard about heart, lung, and blood forms the bulk of what you really need to be successful in Catalyze. Next slide, please. What I thought I would do with this slide is tell you just a little bit of the history and why it was important to establish this center. After all, we've had 70 or more years of heart and lung and blood and sleep research. So why was it necessary to establish a Center for Translational Research and Implementation Science? The picture on the left is from one of the retreats that the NHLBI leadership and staff had back in 2013. And those retreats led to the establishment of CITRIS for the simple reason that there was a real need to have a strategic focal point for completing the translational work. Catalyze is to accelerate translation, but there is that last mile challenge where you take all the work, the preclinical work, everything you've done to clinical studies, and help turn all of that into real world benefit. And so the real focus for the center is the late stage translational research in red and highlighted here, the implementation science, the barriers and facilitators that either stand in the way or help turn the discoveries into real health impact. Think of our domestic challenges in the United States, but also many, many areas abroad. And in that effort, be able to address the health inequities that we see and also support research training and career development. Next slide, please. This slide is an overview of what our key priority areas are. And as I mentioned, health inequities research across all of Heart, Lung, Blood, and Institute is important. And we do that not just domestic US, but we also support global health research across the heart, lung, and blood and sleep areas. But the real core of what we do is what's highlighted in the blue oval, is our implementation science, the barriers and facilitators from getting the innovation to real world settings on the street. And I mentioned the other areas of our domestic and global perspectives, but also research training, career development, and inclusive excellence, ensuring that the groups of individuals that are underrepresented in science get the support they need to also excel, a major priority for the Institute. Next slide, please. This is a cartoon to highlight how the Center for Translational Research and Implementation Science complements the work that Catalyze is doing, and why even this early, as you think of your programs and your applications, it would be worth it to think of the link with individuals with expertise in that area. If you just do one click to highlight one aspect on the slide, please. Great. So all of Catalyze is really going from fundamental discovery science, how we translate from bench to bedside, how we translate to patients, and how we get ready for impactful clinical trials. But where we focus in our center is the late stage T4 translational research, and as I've highlighted at the very top, biomedical and socio-ecological model, where we begin to think beyond individuals into the society in which they live, the health policy implications. In thinking about all of these, your innovations and your creative ideas would have much, much better opportunity for really transformative impact. Next slide, please. So I'll share with you 3 contexts that will make it easier for you to think of the priority areas for our center that can really help enhance your application. And the first is, of course, we think of individuals, whether we think of diagnostic, preventive, therapeutic interventions, drugs, devices, whatever it is. We begin with the individual, but we want to highlight the important context of the communities within which individuals live, the community partners who can be really very helpful to us, as well as community engaged research. A huge lesson we learned during the COVID-19 period is that you can come up with the most outstanding, most powerful drug, but if people don't trust you or people don't believe in it, it will be of no benefit. And in fact, one of the major things that we ended up doing right after we found out we have efficacious vaccines, yet fewer than 4% of African Americans wanted to participate in any clinical trial that had to do with a vaccine. Fewer than 8% of Hispanics wanted to participate. We couldn't even count when it came to American Indian, Alaska Native. So you get the point that while we do the research and come up with innovative ideas, we also have to think of the social and environmental circumstances within which that research is done and bring the community along. So having expertise on your teams that can think ahead as to what some of the barriers might be will be extremely helpful. A second context, the next click, shows that we have to have a focus also on underserved populations. Very often these are populations who are disproportionately impacted by heart, lung, blood, and sleep diseases and disorders. We learn from the polygenic risk modeling exercises that you can actually have outstanding polygenic risk scores that, if applied today, would actually make disparities much worse than they were before. Why? Because they may perform much, much better in one group and perform poorly in the other group. But if you're thinking about these diverse populations and their needs, then your innovation and your creative ideas much more likely to have bigger impact. The final click will bring in the third context, and that is, as we go beyond our own borders and think of low and middle income countries, it's important to begin with what the people there themselves think of their needs and their priorities, not so much what we consider to be our priorities that we bring there. And it's not just abroad, it's also low resource settings within the United States that we want to make sure we prioritize as you think of these ideas. The images on the right are readily available, and we have the citations that can help. The top left is the document that NIH and CDC with ATSDR, as well as the CTSA, is put together as the core principles of community engagement that can make your early translational work even more impactful. The top right is the socio-ecological model, highlighting that, yes, we work on the biology of the individual, but we have to fully understand the relationships individuals have in the communities and the societies within which they live, and also think of policymakers and the policy environment and how these can make things much, much easier. The bottom image is from the most recent report from the National Academy of Sciences that puts community engagement at the center of a series of activities that can really lead to transformative change through systems level changes that stem from the work you're doing, going from fundamental discovery, early translation, but also going the very, very last mile. In the next click, I think we talk about, just waiting for the cue for the next slide, that should help. This is just a cartoon I put together, and if you begin on the right in green, all the wonderful work you're doing is to help with the most important clinical outcomes, increasing the years of healthy life, high quality life, but also promoting health equity. And we want to take those lessons into how you begin on the cartoon on the left. That is at the very bottom of the ovals, all your great ideas, the interventions you're thinking about, the tools, the devices, the therapeutic and preventive strategies, but all of those beginning with individuals, but also thinking about how healthcare providers within the systems they work, we can make things easier for them to be more effective, but also the communities in which people live, and we've mentioned policy and legislation. In the center is critical, and that's the piece about the frontier where our center tries to work with investigators to address. We all know that there are already a lot of high-value interventions that are seriously underused. In the United States today, only about 50% of the guideline-recommended treatment, preventive, diagnostic strategies are used. The vast majority of patients don't get what is recommended or ideal for them. We need to do better than that. At the same time, there's tremendous overuse of very low-value interventions. We need implementation science to tease these out. And so my suggestion to you is, as you listen to what my colleagues shared with you about the terrific biology available in the heart, in the lung, blood, and sleep areas, we also want you to add some thoughts about how we really cross that final mile to get your innovations into real world setting. It has to do with the devices and the creativity and the innovations you come up with acceptable to these communities. Are they appropriate from a cultural perspective? Will they be adopted and sustained? There is a science for studying that, and it is never too early to think about those as you put in your application together. I think that I have one more slide, and then we can break and tackle the Q&A. So if you go to the next slide, please. I don't see it change. There we go. And this is to let you know that we are very, very eager to provide you the support you need from an implementation science perspective. These individuals, the executive team, we're available to you We also have program officers who I couldn't put all on here, but you have these slides, you have the names. Please reach out to us, and we'll be very, very supportive, more than eager to assist you in providing the technical assistance you need to write extremely meritorious applications. I think that's it. In the final slide, I show you very much like Dr. Rizwan shared with you. We are active on Twitter. Very often when opportunities come up, we tweet, and so please follow us. That's our official NHLBI account, and it's NHLBI_translate. Thank you very much, Mike, for inviting us, and delighted to join the panel in addressing some of the questions. Thank you.
Great. Thank you, George, and to the rest of the speakers for spending some time with us today. So I'm going to hand it back over to Marissa now to go over the Q&A questions.
Great, thank you so much. And actually, Shane will be moderating our Q&A for us today. So Shane, would you like to read out the first question?
Sure, happy to do. The first question is, what aspects of blood vessel research and device engineering would fall into DCVS, and what aspects would fall into DBDR? It seems there might be some overlap.
I can take the first stab at that. So I think it really depends on the focus of the work. So for example, for devices, if the focus is on the device development, then it would be likely to be in DCVS. If it was on coatings to make them more hemocompatible, then that's where you start to get into the sort of gray areas. And I think, you know, if the focus is on the mechanisms of, for example, platelet activation, that would go to DBDR. If it's more on the material itself, you know, some sort of nano coating, for example, that would probably go to DCVS. And similarly, I think for the blood vessels, if the focus is on the vessel itself, that's probably going to be DCVS. But again, if it's the activation of coagulation, then that would be more likely to go to DBDR.
Yes, I totally agree with Dr. Buxton. He has explained it nicely. Usually when there is a gray area, when a proposal is in a gray area, we talk to each other. Like I go to my colleague at DCVS and they come to me, we talk, we discuss, and we also invite the PI. It all depends on your experimental design aims. So thank you for the question.
Yeah, and just as a reminder, when you reach out to us, if you already have a 1 to 2 page executive summary of what you're trying to do, that really helps me try to identify who to invite to the call, even if it's across multiple of the different divisions.
Great. Our next question is, if someone has started or works with a company, does that help or does it disqualify the investigator? I assume from Catalyze funding.
So Catalyze is open to all US-based for-profit and nonprofit entities, both, universities, startups, large companies. It's open to any US-based organization. So if you've already started working with a company, that's fine. It's just a matter of, the team is going to be reviewed as far as having the qualifications to execute on the study.
Great, thank you, Mike. Our next question, will the NHLBI interact with other parts of the NIH, such as NCI, in the valuation and funding of these projects?
So Catalyze is specific to NHLBI. So if you apply to Catalyze and you're not within the heart, lung, blood, sleep area, if you're more cancer focused, it will most likely be rejected, and then you'll be recommended to reach out to the NCI for funding. Again, coming to us before you apply and getting our feedback will help you, the investigator, and we can help you identify the best funding mechanism for your studies that you're proposing.
Thank you, Mike. Another question probably directed towards you. One of our currently funded investigators asks, as a recipient of one R33 Catalyze grant, am I able to apply for another Catalyze grant and in the similar field of research to develop a device that will move our product?
It sounds like Probably yes, but that's worth a follow-up call with me so we can discuss it in more detail.
Great. And then a more general question, again, from one of our funded Catalyze investigators. While I understand these to be funding priorities, is there some expectation that these will turn into actual focus RFAs in the future?
So one of the reasons why I invited my colleagues here to this webinar is that the NIH and NHLBI publish notices of special interest for other types of funding announcements. But given the special nature of the Catalyze program, we do not publish notices of special interest. So if there's a specific area that was mentioned today, you can look out in the Federal Register for notices of special interest, or in some cases, there will be future funding announcements targeted for these different areas, priority areas.
Great. And one more quick question. Will CITRIS collaborate with ARPA-H?
I don't believe I see that question. So could you repeat that again? Will CITRIS collaborate with?
ARPA-H.
Obviously, we're learning more about what we'll be doing with the ARPA-H, but in general, the answer to collaboration is always yes, whenever feasible. But very much like Mike said, you have an idea, please reach out to us and we would explore.
Great. Thank you, Dr. Mensah. And as we approach the top of the hour, I'm not seeing any additional questions in the Q&A or the chat at this time. So I will once again thank our wonderful presenters who really did such a great job of building off of each other and connecting the information delivered, but also distinguishing their division's unique priority areas. And even as part of the Catalyze program, this is the first time I've heard a lot of this information, so it was particularly exciting for me. And as we wrap up, Shane, go ahead and launch that evaluation poll. I encourage all of you who are still on the call to submit that poll. It's only 5 questions and will take less than a minute to complete, and really does help us shape and craft our future webinars. And likewise, if there are any specific topics or topic areas that you want to hear more about in upcoming webinars, please feel free to type those in the chat. And finally, I want to remind you all of our next Catalyze application receipt date that is coming up on November 21st. And for additional resources to help guide you through that application process, you can visit our website, nhlbicatalyze.org, specifically focusing on the FAQ page as well as our events page, which has pre-recorded webinars that will help you with that application process. And for any specific questions, you can always reach us at catalyze_info@rti.org. And again, these slides will be posted after the webinar, as well as the recording. And thank you all again for joining.
Great. Thanks, everyone.